Today's lecture is going to focus on the muscles, and here I have the primary objectives that I want to cover. So we're definitely going to look at in-depth about neuromuscular junction transmission, all the neurotransmitters that are involved, in muscle contraction. We're also going to talk about the mechanical action of the contraction of the skeletal muscle. So initially, you will have the action potential that causes the electrical signal. Then you have the chemical impulse transmission, followed by actual mechanical contraction of the muscle. So all that I will explain in detail because it's very important. Why is this important? It's important later on for pharmacology. So there's pharmacology-specific medications that act on neuromuscular junctions in terms of blockade, right, or potentiation. There's also autoimmune diseases that you're going to learn, and toxins, for example, MS, right, or things like botulinum toxin and how they affect the neuromuscular junction. So that will be the predominant focus for this lecture. There's certainly going to be material that outlines, names of specific muscles and some of the basic locomotion and synergists, agonist, antagonist types of stuff. I'm not going to delve into specific details about that. You could learn about that by reading the chapters, but this stuff I think is very important to outline because there's a lot of pharmacology and disease processes that are associated with this. So some of the functions of the skeletal system, or I should say the muscle system, right? So it produces movement, right? You walk, you employ the muscles. We also talked about, right, in the respiratory lecture, the use of intercostal muscles, the diaphragm, how it's employed in the body in terms of utilizing its connections to the to the skeletal system to essentially move the ribs in order to facilitate inhalation of air. It also supports your soft tissues. There's sphincters in the body that guard body entrances and exits. It maintains body temperature. So, for example, you may shiver when you're feeling cold, and it stores nutrients there in form of glycogen. And we're going to talk about all these things here. So the muscular system, as you see here, its predominant 44% that makes up the weight for you. And here you see the other components. We talked about the skeletal system, which is about 20% in weight. Integumentary system, we talked about, this is the skin, and it's about 16%. So the bulk of it is certainly 44% being the muscles that you have. And yeah, of course, it will be different from person to person. Males have more muscle as opposed to females. In addition, someone who is working out in the gym with hypertrophy of the muscular system, they'll have a bigger percentage of that. Right. So this is a cross section of the arm. We talked about different compartments, but here you see essentially these are the compartments we talked about before. You see the blood supplies with arterial and venous supply, and they go deep into the muscle tissue to provide nutrition, right? And we talked about the hemoglobin function that carries oxygen to these tissues and that floats at the tissue level. So certainly muscles need nutrients. They certainly need oxygen that's carried by hemoglobin. And muscles themselves have this storage form of a myoglobin, which we get to later, which essentially gives the muscle that red color. When you go to the butcher and you buy, let's say, steak, and it has that red color, that's because of the myoglobin content. And myoglobin is a monomer; it binds oxygen, and that's why it's red. It's not the blood; it's the myoglobin that gives it the red color. Right. So we're gonna delve in depth about some of these things here. Muscles, as you know, are deeply supplied with blood. So here we see the blood supply to the muscle fibers. In the lecture on skin, I talked about when we are performing intramuscular or IM injection. Why is it important to select at least 1 inch needle, right? Is that you want to penetrate into the muscle. You don't want to be in the fat why? Because the muscles have this blood supply. And the blood supply is what essentially is going to propel your medication to go systemically. It's going to circulate, through the heart, and then it's going to be delivered to all the tissues in the body. So hopefully that medication will, exert its effects. Fat, on the other hand, is not so greatly supplied with all this blood as you see here, right? So it's very important to understand that. Now, we talked about the respiratory system, and here I have especially the diaphragm, and this contracts right before we take a deep breath. So the diaphragm, the muscle that you see here, it will contract, it will flatten out, and this will cause increase in size of your thoracic cage so that the oxygen can be coming in from the atmosphere from high to low pressure. So the diaphragm contraction of the muscle itself, right, facilitates expansion of the lungs and decreasing in the thoracic pressure so that the oxygen can come in and diffuse through here. And we also see the muscles of the intercostals, right? And they are essentially connected to the bones, and they facilitate that bucket motion and pump handle motion. So muscles assist in breathing for you, and we talked about specifically about that. Here we see components of agonist and antagonist. So agonist will be essentially the prior mover or the prior muscle that's being contracted. So here, for example, if we have the biceps and it's the primary mover, that let's say if I'm contracting my muscle, that's what's being contracted, being the primary mover. The antagonist being the tricep muscle, it's behind it, and it has to relax for the prior mover to contract. So antagonist will be right, in opposite function of the primary mover. The same analogy can be made with your quadriceps and your hamstring. So hamstrings will be like the bicep, it's contracting, and the quadriceps will relax, being the antagonistic muscle. And we also have synergists. Synergists, they assist with the primary mover. So they, for example, they can stabilize the deltoid. So here it's a fixator here. And we have the pronator teres muscle, can also be a synergist. So the way this is essentially laid out, the primary mover is what facilitates the primary function. And what you need to understand is for that to occur, the antagonist must relax for that to happen. So here we see, right, the triceps bronchi being in relaxed position. The names also mean something, right? When you hear bi, mean it's double, tri means three. So biceps usually have 2 heads and triceps have 3 heads. So the names of them also have relevance in terms of, why they named like that. Here we see a more close-up view, right, of the primary mover, the bicep, right? And here we see the antagonist being the tricep in the back of the arm. And this has 3 heads. So triceps has 3 heads for tri, and bicep has 2 heads for being the bi. And the synergist will facilitate and assist with the primary mover. And this, if I took your bicep, like I took the bicep muscle, and I essentially wanted to view the entire portion of it. So we see that the muscle is connected via tendons to the bone. So here we have the humerus, which is the long bone in your upper arm. The tendon of the bicep will be connected to it directly. And here, if we basically look at the cross-section area, they kind of show you, right? We have the actual muscle that's coming out. And then as you go deeper into the layers, you see the fascicle that's coming out. We're going to talk about in depth about that. And if we pull out this portion here, right, we see the muscle fiber or the muscle cell. And in it specifically, we have the thick and thin filaments. This we're going to talk about in depth, right? Thick and thin filaments have other names to them also, right? So thin filament will be the actin, and the thick filament will be the myosin. And we're going to learn that cross-sliding of myosin and actin filaments across each other, that what constitutes that movement of the muscle in order to facilitate contraction. That this is the sliding filament theory. If you read in the book, what do they mean sliding filaments? The filaments they refer to as the myosin and actin sliding on top of each other to contract or shorten that muscle. So we'll take a look at that very much in detail. The other thing I want to mention here, sometimes you will see sarco, the word sarco, or sarcoplasm, and you will see myofibrils. What does that mean? Sarco is means muscle, and that's in Greek. And myo also means muscle, and that's in Latin. So they pretty much, the prefix of them means the exact same thing. So sarco just means muscle in Greek, and myo means muscle in Latin. So whenever you see these names here, right, sarcolemma, right, or you see myofibrils, all they refer to is the muscle as the prefix for that, right? So this is just the structure and the cross fibers that I showed you here earlier, and we're gonna talk about more in depth about that sliding filament theory. We're gonna talk about all the neurotransmitters that are involved in this to occur. So this is a more close-up view, what we're gonna delve in. So this, as you see, right, sarcolemma. So sarco means muscle, and myo, right? You see muscle. So both of them, the prefix refer to the muscle. Some important structures that I want to point your attention to here, right? We have the mitochondria, and mitochondria is going to supply the ATP for the contraction to the muscle. This portion here will have the T tubules, which essentially will propagate electrical signal to the sarcoplasmic reticulum that releases calcium. And this calcium is what's going to allow for that actual contraction of this thick and thin filaments. We'll definitely talk about that in depth. I'm just giving you an overview for right now so that you see. The other important thing, skeletal muscle is striated. So we see these striations, and usually these striations is that actin and myosin filaments that you see. Muscle tissue is also multinucleated, and you see this nuclei being present, right? The two tissues that have nuclei in them, we'll learn about the cardiac muscle, which is striated. We'll learn about the regular voluntary muscle that you contract, that's pictured here, the skeletal muscle, that also has these nuclei, and it has all these striations, right? We definitely, once we talk about that filament theory, you're going to learn about all this zone and overlap. You will learn about all these M lines, H bands. So I'll get to that. I'm just giving you a bigger picture overview, right? So we'll, in the prior chapter in respiratory, we'll learn about the hemoglobin molecule, right, that you see pictured here. So I said in that setting, right, it's a tetramer. It has 4 monomers that are basically connected together, and oxygen binds to the heme group, right, to the iron specifically, and this will essentially move the oxygen through all the tissues in the body and offload. But muscle themselves, the muscle, they have myoglobin, right, which you see pictured here. Essentially what myoglobin is, it's a monomer, right, monomer. And oxygen binds to the iron here, and it stores it. And I'm going to show you, right, what's the difference? What's the main difference? So the biggest difference is that myoglobin wants to hold tightly onto oxygen, but hemoglobin has this property where it can transition between tense and relaxed state to offload oxygen needed for the tissues. So here we see, right, the actual representation of the myoglobin. We see that heme group in the center, right, which is essentially iron, and that's what binds oxygen, oxygen being diatomic molecule in this pocket here, right? And if you look at the graphs, I specifically was explaining to you right shift, left shift when we learned about hemoglobin specifically. So if this is your normal hemoglobin, right, as you see the graph pictured here, it has that sigmoidal shape. And what you notice at this portion here, we said it loads oxygen in the lungs, and at this portion here, it offloads in the tissues. But what you notice about the myoglobin graph, is it left shifted or right shifted?
Left shifted.
Left shifted. Yeah, left shift. And what do we know about left shift? Does it want to hold to the oxygen tighter or looser?
Tighter.
Tighter, right? Exactly. So, and you could see, right, if I were to draw a line, let's say at saturation of 0.6, and then we go down, right? You see that I got to reach much lower partial pressure in order for myoglobin to get rid of its oxygen. So truly speaking, when will myoglobin release its oxygen is like in dire circumstances, like where you depleted all your stores. So let's say you're being chased by a big tiger who is going to eat you, right? And you're running away. So first the hemoglobin is going to be used cycling through the blood, right? And the myoglobin stores are only going to be implemented as you deplete all your stores. And it just wants to hold and bind that oxygen very tightly. That's why it exhibits that left shift on the curve. It also has this shape, right? The hemoglobin has the sigmoidal shape. So you see the shape of it like this. Why? Because as more oxygen binds to it, it allows the next molecule to bind more easier and easier. But myoglobin does not have this shape. It has this hyperbolic shape to it. And this also shows you, right, P50. P50 basically means that what percentage, 50% of it is saturated. And at P50, it's 0.26 kPa, essentially, the pressure point, right? And this, right, means it holds to the oxygen much, much, much stronger, right? So it doesn't want to float. So that's what you want to remember. So myoglobin, it's a monomer. It's found in your muscles. It holds to the oxygen much, much tighter as opposed to hemoglobin. It also is there as a last ditch resort. It only will offload, right, in dire circumstances. It also what gives your muscles or red meat, for example, that red color. It's not the blood, it's that myoglobin content, right? So now that we learn all this, we're going to learn there's 3 different types of tissue that you need to be familiar with. There's the skeletal muscle, there's the cardiac muscle, and there's also the smooth muscle tissue. So the skeletal muscle and the cardiac muscles, they are striated, right? They have these striations, which is essentially actin and myosin. And the smooth muscle tissue, which found in, for example, your intestines, right? Your blood vessels, they are not striated. They are not striated, right? And we're gonna show you more in depth, distinctions between skeletal, cardiac, and smooth, right? So as I just explained to you, skeletal muscle and cardiac muscles all have striations, right? They have these sarcomeres, which is composed actin and filaments. The other distinction is skeletal muscles has hundreds of nuclei, as opposed to cardiac muscle, it only has 1 or 2 nuclei. The smooth muscles, right? They have 1 nuclei, uninucleated, and they do not have any striations, no striations present, right? You see these are what the striations shown, and the smooth muscles does not have it. And primary location for them is going to be GI tract, your intestines, your blood vessels. What's the function, right? So skeletal muscles, besides the locomotion, right, that is done, and the voluntary movement of muscles, they also aid in circulation. So if I squeeze my muscle, that facilitates the flow of blood in the veins, because veins, as you know, they have valves, right? And that what will propel blood to go up. And when the valves close, that will not allow blood to come back. So if I squeeze my muscles, that contraction of the muscles will allow movement of blood in the veins, hopefully coming back to your right heart and then circulating through the heart. The other important thing we're going to talk about is skeletal muscle is under voluntary control. So I can control if I want to contract my muscle or move or pick something up, right? I can, if I wanted to pick up this pencil, I can do so, right? And that's done via somatic system. Cardiac muscle, on the other hand, you cannot really control by your by you wanting the heart to contract. So heart contracts in your body under involuntary control. Yes, you could speed it up by specific neurotransmitters that are released, right? We're going to talk about more of that when we do the cardiac chapter, but the heart is contracting through your body consistently. Same thing if after you eat your food, your GI, your intestines, they start the process of peristalsis, where they churn and they move the food through the small intestines, large intestine. You cannot control that by your willpower, right? It's done by the your autonomic nervous system, and we're going to talk about that in depth. Also, they have Gaba junctions, right? So the Gaba junctions in skeletal muscles, each of them is isolated, but we're going to learn in cardiac muscle, there's special Gaba junctions that connect each myocyte to each other, and that propagates the electrical signal and to go through them. We're going to see these discs called intercalated discs present specifically on cardiac muscle that have these Gaba junctions that allows myocytes to connect to each other, right? And this we already talked about. Skeletal muscle is under voluntary control, and it's done via somatic nervous system. And the primary neurotransmitter we're going to learn in depth about is acetylcholine. The heart is, and the organs, right, such as GI tract, they're under involuntary control. They're controlled by also autonomic nervous system, right? And here we see that autonomic nervous system. Myogenic, what does that mean? It means in the heart itself, you will learn there is these special structures called the SI node, AV node, and the bundle branches that propagate the electrical signal through the heart. So heart in itself has its own conduction, right? SI node, AV node, bundle branches, and it's myogenic, so it can contract in itself through the propagation of these systems, right? So this is just a close-up view. This is gonna be that essentially the sarcomere, or the contractile portion that we're gonna spend a lot of time talking about, right? So here we see the sarcolemma surrounding all of these units. All together, they're known as the muscle fiber. The mitochondria is here. Why? Because it supplies the ATP, right? And we need that ATP there in order to power that muscle contraction. If you take out right, one fiber from here, it's called the myofibril, and myo, we said, means muscle. So it means muscle fibril. And here we see these Z discs. The shape of it is a Z, right? And if I basically bring out what's in the middle between these Z discs, you see this configuration here. So everything that's located in this part of the myofibril, if I basically zoom into it, you'll see all these components. So what are these components? So we have the actin filament, which is this thin filament. We have the thick filament, which is the myosin, right? This is elastic filament tighten. This is allows to spring, once it contracts, to allows it to spring back. And here it's essentially the zones where it overlaps, right? They will create these dark bands you will see on the tissue. So where actin and myosin overlap, they create dark zones. Where they do not overlap, they create these light zones. And we're going to talk about what causes the actual contraction of these thick and thin filaments as we proceed forward. Right?
Quick question.
Yes.
Can you do that one more time? What's the purpose of the actin filament?
Yeah, we're gonna talk about this in depth. This is just a basic overview to give you an idea what we're talking about. But the purpose of the actin filament and the myosin filament, the myosin head that you see here is going to bind onto the actin, the thin filament, and then it will contract, causing a power stroke to basically move these filaments towards the M line or the middle, right? Causing contraction. I'm going to talk in depth about how this is done. This is just a like overview, so you have an idea how this process works.
All right, got it.
All right. This is what I was explaining to you. You know that veins have valves in them. Especially, we were learning this for those of you who came for IV cannulation or IV access. I was explaining to you, right? You want to pick a vein when you start your IV, right? You want to have it straight. You want to make sure, right? It's not, moving in this form, right? Where it's zigzagging, and also it has valves. So what may happen is you may put your catheter right at the tip of the valve, and the valve may prevent, right, the flow of fluid or whatever you're giving. Muscles here, right? When you contract, they will essentially what will propel the blood coming back. Why? Because as the muscle contracts, the valves open and they move, right, the blood towards the right heart. Here, specifically, what we're going to focus on for the musculoskeletal system, right? We're going to talk about the somatic system. overall, your nervous system is subdivided in two categories. We have the central nervous system, the CNS, which is composed of the brain and the spinal cord. We're going to talk in depth about that in the neuro lecture next week. But today we're going to spend some time talking about the peripheral nervous system, specifically the somatic system. How is it divided in depth? I will show you some slides. This is an actual cardiac tissue. It was from the slide. Why do I have it here? I want to show you these intercalated discs. And I said the intercalated discs are very specific to cardiac tissue. They're not found in muscle. They're not found in smooth muscle, right? So these are only found in cardiac tissue. What do they do? These intercalated discs that you see the arrows pointing. So first of all, they have desmosomes. So what are desmosomes? Desmosomes are essentially, they anchor the tissues and these cells, the myocytes, together. And in addition, what these intercalated discs have, they have something known as these Gaba junctions. And what Gaba junctions do, they facilitate ion exchange. So ions being, sodium, calcium, right? So they propagate from cell to cell in order to basically move that ion potential to the next cell, so they depolarize together. And this is done under involuntary control, specifically the myogenic control using the SA node, the AV node, to essentially conduct that. But this is why I have this here. This is very specific to the cardiac muscle. The other tissues do not have this. So remember, intercalated discs, they have two components. They have the desmosomes that basically keep the cells together, right? They prevent them from going apart, separating. And they also have these Gaba junctions that allow ion exchange to occur. And this is a colored view, same tissue, cardiac muscle tissue. This was from actual tissue, like a real tissue, not like a picture it was taken from a microscope. So you see this, the nuclei, right? So we said there's 1 to 2 nuclei with the myocytes, and we see them pictured here. And we see the striations as well, right? This is a picture of a heart. This is not a human heart. This was, I believe this was a pig's heart. And what I want to point your attention to, right? You see, this is the left ventricle. This is the right ventricle, right? Left ventricle, right ventricle. And you notice the extent of the size of the left ventricle, how much hypertrophy this as opposed to the right ventricle. And that's because the right ventricle propels blood to the lungs, lower pressure, but the left ventricle propels blood to the entire body, right? We know normal blood pressure being 120/80. So this left ventricle has to overcome 120 systolic pressure in order to eject blood through the aorta, right? The aortic valve. And we see the hypertrophy that this sustains, right? So this is just showing you the valves in a more in-depth picture. But the main takeaway, right? This is what I want to point your attention to, the size difference between right and left ventricles in terms of, and why is this important? The functionality that it serves, right? This is the pulmonary circuit, and this is the systemic circuit. The other thing is, this was from the human heart. If you unravel all these muscle fibers, right? They took the heart and they basically unravel it. And why is this important? This is important when the muscle actually contracts, the way the actual blood that's being propelled, it has like this churning motion. Think of it if you did laundry by hand, right, in your tub, and then you want to wring out the excess water. And what you would do is you would take that laundry, right, and you would like wring it out. And that's how heart contracts, right, utilizing all these fibers of the muscle in order to allow blood to move and go systemically. So you see how the fibers are rolled together, and this churning motion will facilitate the motion, right? So this is the subdivision of the nervous system that I was talking about. So what we are focusing on today, we're going to focus on the peripheral nervous system. The peripheral nervous system divides into the sensory, right? You feel the sensations, and then you have the motor response, the effector response, or the efferent division. That's why when you are doing your trauma assessment, we check for circulation motor sensory, right? Because you may have damage to the sensory, but the effector muscle, right, the efferent muscle may still be functional. or it may be the other way, right? The sensory may be damaged, or the muscle, the motor may be damaged. So as we go from the motor division down, right, we talk about the somatic nervous system, which will dive in depth, which controls the voluntary motor motion. And then we have the autonomic nervous system, which is not under your control. This is under, right, involuntary control. It's done by neurotransmitters. And this controls your heart, right, your blood vessels, controls your GI peristalsis. But today, what we want to focus on, and I want it to be very, very clear, we're going to talk about the somatic system, which is under voluntary control. And the primary neurotransmitter that's responsible for that will be the acetylcholine. And we're going to talk about how it's produced in the body, how it's made, and how we employ it, right? So just to recap, the divisions, right, being central nervous system and the peripheral. We're focusing on the peripheral. And as we go down, we're going to go down to the motor, the efferent system, which is further subdivided into somatic and autonomic. And today is the somatic system that we are focusing on. This is just a schematic view of exact same thing. And just to point your attention, right, we have the efferent nerves of the motor system, which is controlling the skeletal muscle. And that's what we're going to be spending our time today. The remainder of this will be covered in depth when we do the neural lecture.